Okay, our last video is the coolest part. It's the part with the most steps. And this is something we haven't talked about before. It's called meiosis. M-E-I-O-S-I-S. Maybe I'll make it pretty. This makes gametes. Gametes are also called sex cells. These sex cells are haploid. They need to have half the DNA so that they can go meet up with another cell that has half the DNA and make a new organism. We'll talk about that tomorrow. So half the DNA of a somatic cell. If you didn't do this, then a human would have 46 chromosomes, and then if you just sent your 46 and 46 came from the other parent, they'd have twice that many. You'd end up with tons of chromosomes. That would not work. So this is still Fred. He still starts out with a cell that looks like all the other cells in his body. Big, medium, small. Big, medium, small. And he goes through S phase. So this part looks the same. S phase. Now I have 6 duplicated chromosomes. Oops, ignore that. And in this case, some really cool and weird stuff happens. So before they divide, my homologous chromosomes are going to hang out right next to each other. So here's my chromosome 1 hanging out right next to chromosome 1. Chromosome 2 is going to hang out right next to chromosome 2. They don't necessarily line up. It's not that red is always on the right. In fact, let's say that blue happens to be on the left over here. So they line up right next to each other. These are called tetrads. So when they form pairs of homologous chromosomes. They actually wrap their arms all around each other and they swap pieces. So remember we saw earlier that there's such a thing called recombination? This is when it happens. Oops, not an S. This is when it happens, is that they'll actually swap pieces. So now this is when the drawing gets complicated. Now I have this chromosome, this one, and I'll put that there because these ones swapped, maybe they just swapped one end of their DNA. And now this little guy has a red one. And over here, the same thing happened. Let's see, oops, medium, and super small. Oh, I'll fix that. So now I have some pieces that look kind of new. And instead of pulling apart these identical pieces, what happens in the first division is that these homologous chromosomes get pulled apart. My phone is getting emails. So my homologous chromosomes split, and I end up with, don't make your drawing too pretty or it'll take you too long. I'm doing it by color. It's a big one, medium one, the big one should be bigger, sorry. And the small one. All right, so I'm now split them in half. This is called meiosis 1, the first division. Then the second division looks a lot like mitosis. The chromosomes that are left in each cell line up so that these are almost identical now, but they're not completely identical because some of them swapped pieces. They line up so that they can get pulled separate parts of the cell. This has a little N. big, medium, small. Medium should be bigger. And then they will get divided in half. So they'll split again. This piece will go this way, this piece will go that way, this piece will go this way, this piece will go that way. They'll split in half. I'm not going to draw that on both sides. And I will end up with two cells that came from this one. So I've got this piece on this left side, and then I have this piece, and then I have this piece. And none of these have any weird spots because they didn't exchange any pieces on the left side. If I looked at the left side, everything looks normal. Then if I look on the right side, those are where all of my recombination happened. So I've got 1, 2, 3. So this one split into two cells. I'll keep adding my arrows. And then over here I do the same thing. I look at what I've got going on. All right, everything on the left side, I split these identical pieces of chromosomes in half so they're not duplicated anymore. This is my big chromosome piece, my medium chromosome piece, and my small chromosome piece. Then over here, if I look again, big piece, medium piece, and the small piece. So now I have gone through, this is called meiosis 2. So there's two divisions, meiosis 1 and meiosis 2. And I end up with 4 daughter cells. The question is, do they look like my parent cell? They look similar, but there's definitely a difference. First of all, my 4 daughter cells, does any one of them look exactly the same as the other? All are unique. There's no one that looks like the other. So I get 4 different cells, which is cool. Secondly, let's go through and do the same thing that we did before. I'm going to ask the question, haploid or diploid? Let's scoot that over. The question is haploid or diploid? And then, are they duplicated?